Okay, great. So thanks, everyone. I'm going to start us off in the presentation and provide a framework and a background of why we focused in this area, as well as some of the assets that we have at our disposal at PRA Health Sciences to be able to address and create a solution for this. So Nathan, if you go to the next slide. So over the past several years, PRA has made some significant investments in the area of data, patient engagement, and technology, as Bev mentioned. Although historically PRA was, we are a CRO, we run clinical studies. However, our leadership team really looked to determine how we can make research better and design better research and help answer scientific health questions in different ways. So we've acquired different patient engagement technologies, and specifically a few years back we acquired Symphony Health. And that really helped enhance our data assets as well as our expertise in this area. Although each of these components have their own kind of distinct services and expertise areas, what we have been able to do over the past several years is integrate these, and you'll see this within the solution that Kerry and Nathan outlined. That not only are we providing solutions, for example, with our Symphony historic commercial visibility and data with tools such as Metis and tokenization across multiple data sets, we also can integrate that with the other assets that PRA has to create a comprehensive multidisciplinary solution, and Kerry will go into a little bit more detail on that. It was really exciting to be part of this team, and when we first started creating this solution, we had a different focus. And with the kind of shift of the methodology for Fuse and our presentation, given the current COVID pandemic, we actually took a step back and said, well, how can we use the tool we created to create some relevant insights in today's environment, specifically around COVID and some of the treatments that are being tested and used, post-marketing treatments. And Nathan's going to go into a lot of detail around what we were able to find, and it was really eye-opening. And I think there could be a lot of hypothesis generation based on what we found, and help focus researchers even more into how to answer the questions to address the pandemic in a safe and effective way. We go to the next slide. So as we all know, primary data collection has been the foundation of the way that we do health research. Collecting new data for very specific research purposes like clinical trials, registry, natural history, and that's what we've had, historically, and it's great. It has a lot of very specific information. We also have a lot of internal validity when we run clinical trials. We have the perfect patients taking treatments in the perfect way, or within a particular protocol. So we have a lot of good evidence that comes out of primary data collection. However, that's just one piece or one layer of evidence generation. So what we really look to do is leverage secondary data, such as claims data, which is what we use specifically for this example, EMR, EHR data, to help enhance that. And that gives us more external validity to the data we collect in primary data, or the evidence we collect in primary data. And it really helps us to have a wider view of what's happening. And particularly in the real world, we all know different types of patients are prescribed and take products in different ways. And how does that really affect the safety and effectiveness of a particular product? And then the third layer we really look at when it comes to evidence around healthcare is complementary data. And we actually had another team from PRA who presented their solution around complementary data, social listening, and how to find particular unknown safety signals. And so these three layers of evidence really give you a broader and more full picture of what's happening within a particular treatment. Go to the next slide. As I mentioned, we had a multidisciplinary team. I just want to recognize and thank the group here that spent a lot of time and effort in addition to their day job with developing this solution. And I really think it's something not only that provides evidence, but it's very user-friendly. And you'll see when Nathan provides the demo, you don't have to be a programmer to use this. And you can get some very meaningful insights with a real simple user face to create your scientific questions. But then also the results are served up in a very easy to interpret and understand, what that's actually telling you. Go to the next slide. Not only did we want to make sure we had scientific validity and structure to our solution, but we also wanted to make sure it was clinically relevant. As we all know, something can be statistically significant, but is it clinically significant? So we pulled in our therapeutic experts. As you can see here, Dr. Fred Lewis was part of our team to really determine, are we heading the right direction? Is this going to be important to physicians and patients and caregivers? And so given the fact of polypharmacy and the fact that, we're moving into a new frontier of different types of treatments for diseases that we never even knew about, this becomes even more important for us to understand. And when we see a particular treatment having some type of safety signals, can we focus in on a specific type of patient that may be more at risk versus others? So it may be a very good treatment for a wide variety of patients, but we want to really be cautious about a certain cohort or a subset of patients. And really that's what we're looking to do, is not just create the solution, but make it something that's meaningful for drug developers as well as medical and health sciences. Next slide. So I'm just going to frame our solution of kind of what are the current methodologies of understanding drug-drug interactions in the life cycle management of a product. And then I'm going to, as I mentioned, hand it to Kerry and Nathan. So as we all know, in vitro is really one of the first places that we are able to test drug-drug interactions, and particularly known drug-drug interactions. And as you can see here, a quote from the FDA that it allows us to continue to move forward with the development of a drug and identify some of the major safety or interactions that we need to be cautious about. But it's only, again, very, very specific research that's done at that point. Go to the next slide. As we move through the pipeline of a product and we start doing clinical trials, as I mentioned, our inclusion-exclusion criteria really help dictate those kind of perfect patients, if you will. So we have a very specific group of patients, and the protocol is going to dictate how they're going to take the product and any other treatments that are excluded. So again, it gives us more insights around the pure safety and effectiveness with a lot of internal validity. But then as we move into a marketed environment for a product, we really have two traditional ways of collecting that. One being spontaneous reporting, which is a great methodology to be able to give an outlet for physicians and patients to report events that may be occurring in a market environment. And then we also have things like post-marketing commitments and post-marketing research that helps give additional framework around that. However, we do have a gap there. We still are missing a broad, diverse population being measured to determine, really more variety of drug-drug interactions that are maybe unknown and occurring. And we actually had a discussion with Dr. Topol. Most people on the call probably know him very well. And this is an area he's very passionate with, and as a lot of publications, been very vocal. So we are excited to be able to provide a new solution to the health research environment to really help enhance the evidence that we have. I'm going to hand it to Kerry to talk about the importance of our multidisciplinary team, and then Nathan will give us the demo and the overview. Thanks, Kerry.
Thanks, Kathleen, and thanks for setting that up for us. So when we started with this challenge from the FDA and Fuse, we started building our team within PRA, and we kept expanding and pulling more people in because we have the access within PRA and the different divisions that we work across to pull and look at data in different ways and consume data in different ways. So to really expand the solution and come up with a great idea and really make it a strong and diverse solution. So, as I said, and as Kathleen pointed out for the team earlier, we have backgrounds in nursing, epidemiology, and therapeutic experience. We have individuals who consume big data and taking a look at things, post-marketing and during the clinical research. So the solution that we came up with, we think is very dynamic, and I won't hold you any longer from seeing what we came up with. So I'll hand it off to my colleague, Nathan.
Thank you very much, Kerry. So our solution really stands on the shoulders of a team of very talented data engineers who assemble a data set that really gives us a commanding view of the United States healthcare environment. We focused on data from February through April of 2020 because we were especially interested in the impact of COVID-19. And so that data set included 160 million patients, over 1.1 billion prescriptions, 640 million diagnosis codes. And then within that larger data set, we paid special attention to some subgroups. We looked at 293,000 patients nationwide who were taking chloroquines who did not have a prior diagnosis for autoimmune diseases like lupus or rheumatoid arthritis, and also didn't have a prior prescription for that chloroquine in the previous several months. We also looked at a geographic cohort of patients centered in New York, New Jersey, and Connecticut, and that's the group of 1.7 million patients that we focused on for our network analysis. So our approach to the Fuse challenge was to create a model of our patients as part of a network graph. And our model has nodes that represent drugs and patients and then diagnoses. And it also has nodes that represent the events that tie a patient to their drug or a patient to their diagnosis. And modeling the data as a graph like this has some technical advantages that we'll talk about later. But the other thing that we felt strongly about is that it really helps us communicate with the various diverse members of our team and the various stakeholders who are involved. This picture is simple enough that both a machine learning engineer and an MD can figure out what's going on. And we know that when we're talking about artificial intelligence in the context of healthcare, that explainability is really essential. And so starting from a graph gives us a good foundation for explainability as we develop our solution. So once we developed our model, we loaded that real-world data for 1.7 million patients, and we loaded into a graph database called Neo4j, and I really appreciated the support that we had from our partners at Neo4j to make this project possible. And again, as we started to explore the graph, we wanted to focus especially on things that might be related to COVID-19. I think this is one area where our approach of looking in real-world data for these drug-drug interactions is particularly useful. There's some fantastic work that complements our project here that has been done to infer new drug-drug interactions that are adjacent to things that we already know about. But in an environment where so much is new and people are innovating so quickly to try to battle this pandemic, we felt like there's probably things that are interacting in ways that are totally different than what we've seen before. And that's where we wanted to focus our study. And we feel like by looking at the real-world evidence, we're going to be able to possibly pick up some things that we might have missed otherwise. So in addition to modeling the data at the patient level, we can use our graph then to aggregate our results and start to look at relationships between drugs and diagnosis. So here in this picture, I have a drug and it's related to a diagnosis, and this line on the left represents all the patients who took this drug and then later on had the diagnosis. And the same thing over here on the right, another drug, and all the patients who had that diagnosis. This purple circle represents the patients who took the combination of the two drugs, and then we can see the people who took the two drugs, and following taking those two drugs, they saw the diagnosis. That's what the center line here represents. And as we applied this methodology to our graph, we saw some patterns that are potentially interesting emerge. We noticed that for patients who are taking either hydroxychloroquine sulfate or chloroquine phosphate in combination with benzonatate, that their rate of tachycardia diagnosis following the combination of the drugs was higher than either drug alone. And so that gives us an interesting hypothesis to explore further. Also, we looked at not only individual diagnosis codes, but diagnosis categories. So this category R00 includes not only tachycardia, but bradycardia and a few other abnormalities of heartbeat. And we looked at that category as a whole. We saw that patients taking chloroquines plus septinir had a higher rate of diagnosis for any of those ICD-10 codes that are part of that category. So that information that we see a higher rate of diagnosis with a combination of drugs is certainly interesting, and it starts our wheels turning as far as what's really going on here. It's important for us to figure out, though, are the patients who are taking both of those drugs in combination at least somewhat similar, more or less similar to the patients who are taking either drug on its own? And this is where the graph can also help us, because we can use the network graph to identify communities of patients with similar prescriptions and diagnoses. And so if you look at this picture, I can see two patients, and they have two prescriptions in common, and then out of a total of 6 prescriptions and diagnoses in the whole picture. So 2 out of 6, that gives me a quantifiable way for me to say how similar are these two patients. And then based on that similarity score, I can use unsupervised machine learning algorithms to discover communities within this big network. And those communities are people who tend to have similar diagnoses and similar prescriptions. And then we can explore that drug-drug interaction, the same picture that we're looking at before, but focus it on that specific community of people who are more or less similar. The other interesting thing that we can look at with this is if I have a community of patients who all share a lot of diagnoses and prescriptions in common, and I see that there's a large proportion of patients in that community who have a COVID-19 diagnosis, it's a good chance that the people who are in that community and have so much in common with them might be potentially undiagnosed COVID-19 patients. And so as we're trying to get a picture of who has been infected with COVID-19, this gives us one potential approach that we can use within our real-world data. So at this point, we're going to switch over to a quick demo of what our tool looks like. I'll stop sharing that screen for a moment. And share over here. So we can search in this tool for top interaction candidates for a drug, and I'll choose hydroxychloroquine. And then I'm going to limit it to a specific chapter in the ICD-10 diagnoses so that I can focus in on a specific area. So chapter 18 is where we see diagnoses related to irregular test results and lab results. So we'll let the graph kind of start to fill in here, and I can see I've got hydroxychloroquine kind of in the middle. And then I see a diagnosis, several things related to palpitations, and then I see this one over here related to tachycardia. So if I just want to hone in on this one, I will select these particular nodes and dismiss the others. And then if I double click on some of these, it will give me some more details. So I see out of this subset, and this is the people who are in New York, New Jersey, and Connecticut, I saw there were 46 4,600 who were taking the combination. I can see how many 345 hydroxychloroquine patients followed up with tachycardia. 1,000 of the benzonatate patients followed up with tachycardia. I've got 20 who are taking both, and then I also see a chi-square from either direction as far as if I started with hydroxychloroquine and added benzonatate or went the other direction, what's that chi-square look like? So we can start to explore the data here, and then as we generate the hypothesis in a tool like this, we can take it further with other further analysis. Let me show a different look that we can use to explore these communities as well.
And Nathan, while you're pulling that up, from a clinical perspective, again, from a data perspective, you can see that there's relevance, but in many cases, we're seeing information on Dr. Google for patients to take, that this combination actually is effective. And again, for a particular cohort or patients with certain confounding effects, this additional tachycardia could be life-threatening. And so again, it really helps us hone in on, where we need additional research to happen to really know what we can potentially treat this pandemic with. And we all know about kind of throwing, a whole product out versus do we really just need to focus it or understand it better? So I think that from a clinical perspective, I think it's something that this can really help us focus in on treatments that may be safe and effective for a wide variety of patients. And I know from a timing perspective, we're getting a little tight. So Nathan, I didn't know if we wanted to just wrap up and see if there's any final questions.
Sure. Yeah, I'll just expand. This is showing our different communities that we detected, and you can see here that there's a way that we can tell which are the important drugs and which were the important diagnoses as part of this community. So that's another way that we can use the tool to explore the data set.
Yeah, it's great. And our PRA is involved in a lot of COVID research right now. So this tool is really important when we're designing protocols. And we also have some future enhancements to this that we have planned. And again, our contact information is on the slides that will be disseminated. So if you'd like to get a deeper dive, or this is something you think that would provide value to your organization or in the research you're doing, feel free to reach out, and we'd love to have a dialogue.